knowledge-based background is on it, but it's a very, very caustic disease on the body. And then on top of it, the therapies themselves are incredibly toxic to the body. And so this is a late stage treatment. So the patient has already undergone other therapies at this point. So being exposed to this level of toxicity really does deteriorate their body and make their chances of survival limited. So that's why, for our overview, the main purpose of this research project is to try to synthesize and evaluate a novel platinum drug, and then we're going to use it on prostate and breast cancer. And further, the goal of this project is going to be to diminish the proliferation of cancer cells while mitigating the damage done to the healthy normal cells of the same type. So going into our methods, first, we broke up the project into two parts. So part one, I mainly did with Dr. Lieberman-Martin. This was the synthesis of the drugs. So the first thing I did was synthesize a pre-existing platinum drug that we had provided by another university. We went ahead and synthesized it under nitrogen-free conditions in a glove box. After we had synthesized the compounds, we then used different solvents to dissolve them. And we characterized what compounds we had created using an NMR scan. So the NMR scan confirmed the identity of the new product, and then it also indicated what the purity of those products were. The second part, this was done with Dr. Marco Pisaffi, was the testing of the drug. So this is where we actually get into the cancer therapy part of it. So the two drugs that we ended up making were NCN-PTCl. I'll refer to it as the platinum chlorine drug throughout the presentation, and NCN-PTCH3. I'll refer to that as the methyl platinum drug throughout this presentation. So what we did was we first dissolved them in dimethyl sulfoxide. This is a very common solvent used for pharmacology. It's a very useful solvent when it comes to looking at biology and about how drugs react with cells. Then we took these two drugs, tested them on cancerous and non-cancerous cell lines from both breast and prostate cancer. And we wanted to look at how these cells metabolize when they were under treatment. We also did some imaging of the cells so that we could see what the morphology of them was as they were being treated. So here is the actual synthesis of the platinum drugs. As you can see, we started off with the chlorine, the platinum chlorine drug right here, and then we went ahead and we made the methyl. So some interesting things about this synthesis and also about this drug structure is these pincer ligands right here. These add a little bit of sterics, but they also do contribute to the drug's reactivity. So our theory was that either the chlorine or the methyl group would possibly react to the cancer cells, but not react with the non-cancerous cells. So that was kind of our hypothesis or our theory. We also have this group right here. So that aromatic kind of group is going to help contribute to the reactivity. And if you go back and look at this compound, kind of a similar structure. So we kind of tried to base this structure off of the oxaloplatin when we were developing these. So here we go into our experimental parameters for the actual testing of the drugs. So a couple things is we used 4 different epithelial cell types, 2 prostate and 2 breast. I'll show you what those look like in a minute. We also did the treatments. Obviously, we did it with the chlorine and with the methyl groups, and we also used a control where there was no treatment. And then we decided, since we didn't really know what the concentrations of these drugs would be, we started at 10 micromolar and 20 micromolar. We actually did do a couple experiments at other concentrations, but the cells were not really showing any reactivity. So the main experiment that I'll be showing you results for was with the 10 micromolar and the 20 micromolar. So these are the cells themselves. This is them with no treatment. This is the control group, and I'll also be showing you these images later in comparison to the treatment groups. RWP1 is the first cell type. This is the non-cancerous cell line for prostate. LNCAP is the second. This is the cancerous cell line for prostate. NCF10 is the breast non-cancerous cell line, and NCF7 is the breast cancer cell line. So in order to do our experiment, first we had to count the cells. This is done using this little machine called CountTest, The cells are counted before and after plating. What we want to do when we are counting them before is we're trying to determine what the concentration of living cells in the plate is so that we can measure exactly how much of a concentration of the drug we need. And then after treatment, we use that as an evaluation to see what the morphology of the cells are when they're done under treatment. So this is the assay that we actually performed. So really quick overview, WST assays are a cell metabolism assay. However, because the cell type that we are using is a genetically modified cell that proliferates pretty much rapidly, uncontrollably, we can actually use this assay not only to look at a cell's metabolism, but also to look at a cell's proliferation. So it's kind of a cool tool to use in a case where you have genetically modified cells. So the process is pretty simple. We go ahead and we culture the cells on a plate. We let them incubate overnight. We do a pre-rating. This is done at 660 nanometers. This is done to normalize our data later so that we can set the controls to 100% and really see what the difference is in the cell's morphology post-treatment. We add our drugs. We do let them incubate for a while. We add the tetrazolium salt. That's basically the WST reagent. I'll probably be referring to it as WST reagent for the rest of the presentation, since it's a little easier to understand. We incubate it for 3.5 hours, but during those 3.5 hours, we go ahead and we measure the absorbance at every 30-minute interval. What this is doing is this is seeing how the cells are metabolizing over time under treatment. So then if you want to look at what the WST assay is at a molecular level, so the WST reagent or the tetrazymal salt is going ahead and it's reacting with the NADH-NAPH pathway, which I'm sure Dr. Owens is very familiar with this pathway. It is related to the cell metabolism. So what this is doing is as it is reacting with this pathway, it's actually changing the structure of the WST reagent to emit a certain dye that is then read as an absorbance, which can be interpreted as the cell's metabolism. So now on to our results. So these are the cell imaging. As you can see, I broke them down into these are the prostate cell lines and these are the breast cancer cell lines. The non-cancerous one, LNCAP right here, you can see when it's being treated with the methyl group versus the chlorine group, the morphology changes quite a bit. You can see that the cells are clumping together. They're not really as spread out as they are under control where there's no treatment, or even under, the chlorine where there's still a little bit of dispersion between the cells. Interesting enough, you also see that the cells are darker. We can't fully interpret that as cell death, but we can also interpret that as the cells are not metabolizing at the rate that they should be. And then for the cancerous cell lines, what's interesting is you actually see the opposite effect. You still do see the morphology change, especially with the CH3, the methyl group, compared to the chlorine group, but you don't see as much darkening of the cells, which can sometimes, again, indicate cell death, but it isn't a guarantee of cell death. And then when you get to the breast cancer cells, you do see a similar trend. So again, with the non-cancerous cell line, you see the morphology, they shrink, they kind of shrivel up. They're not as dispersed as they are under the control or even under the chlorine. And then kind of the same issue with the cancerous cell lines, but to a limited degree. There's very little differentiation between the chlorine and the methyl for the cancerous cell line, which you'll see a little bit later when I show you the statistical results. But yeah, overall, the morphology is indicating that the non-cancerous cell line is actually being treated more rapidly than the cancerous cell line. And the graph also affirms this. So as you can see, for the cancerous cell lines, they're being proliferated a lot higher than the non-cancerous. Interesting enough, the 10 micromolar area is what is most important, because at the 20, you see that the proliferation is just kind of all down. So pretty much everything is being killed at that high of a concentration. But when you do it at the 10, you start to see a little bit more of teasing of results. And as you can see, we set the control to 100. This is also a percentage. So this is not like the absorbance values themselves. This is actually the absorbance values, the averaging of each of the plating and then percentage. And so what are the conclusions that we can draw from this? So the first big conclusion is that these platinum drugs do decrease the proliferation of both prostate and breast cancer cells. However, there are a couple limitations. Some things that we did find is that the ideal concentration is at 10 micromolar. Unfortunately, this is not physiologically possible because that is a very high concentration for what is normally seen for drug therapies. Additionally, we found out that the methyl substituted group was the one that was appearing to affect the cancer cells more than the chlorine group. Our limitation is, of course, the platinum drugs are not differentiating between the cancerous and non-cancerous cells as we had hoped, and in some cases are actually killing the non-cancerous cells more than the cancerous cells. So our final conclusion is that while the cytotoxicity of the drugs may not be viable, there is still significant effect on the proliferation of cancer cells when they're under treatment. And so based on the data, it's concluded that these novel platinum drugs could be used as leads for further targeting drug development in prostate and breast cancer cells. And then this is a thank you to both of my mentors, Dr. Basafi and Dr. Learman Martin. These are my citations, and thank you. And then do you guys have any questions? Good.
Do you expect that the mechanism of action for the drug will be very similar between the breast cancer and the prostate cancer cell line?
Yeah, I would interpret that the mechanism of action will probably be the same. Part of the reason why we wanted to use 2 different cell lines is we did want to see if this was specific to one type of cancer or if this could be applied to multiple types of cancer. So since there wasn't really a differentiation between the 2 cell lines, I would assume the mechanism of action is the same.
Can I ask a follow-up? So what is the drug actually targeting in the cell?
Yeah, so what the drug targets in the cell is it actually targets a specific pathway, mainly the PKA pathway. It also does have some effect on the cell's metabolic rate, but mainly it's used to just kind of kill the tumor. So it's not really meant to do like specific pathway destruction as much as it's meant to just kind of in general kill the tumors. It's a very, very toxic drug, so it really is like a last resort drug. And because of that, a lot of times, the pathway morphology is important, but it's mainly just sort of a last resort killer of tumors. So. Any other follow-up questions? Thank you very much.
It's a pleasure to introduce Doris Yang. She was actually my supplemental instruction in my first semester here at Chapman, and she was a great SI. Today, she's going to talk about her project assessing the downregulation and inhibition of androgen receptor activity in triple-negative breast cancer using apigenin. And her advisor is Dr. Marco Bisafi.
Hey, hi everyone. My name is Doris Yang. I will be talking about the downregulation of the androgen receptor in prostate and triple-negative breast cancer using apigenin. So what is the androgen receptor? This is a nuclear transcription factor that plays a large role in mediating dihydrotestosterone. This is an androgen. It's a natural ligand to the AR. And what happens is that the ligand, which is our DHT, will bind to AR to control AR function. And it happens by acquiring a signal through physical ligand binding, in which it will initiate many things such as protein interactions and also regulating gene expression. We care about the AR because it is heavily linked to prostate cancer. It has been shown previously that a decrease in AR expression will in fact prevent the progression of prostate cancer. And the novelty here is that AR has now been recently discovered to be linked to triple-negative breast cancer. This is the most negative or the most aggressive type of breast cancer. And so using this connection, there may be developments in like drug treatments and everything. So we wanted to expand a little bit more on that. I have here just a little bit of a pathway to show you. Basically what happens is androgen, this molecule right here, usually it's testosterone. It will be converted into DHT, which is our natural ligand. It enters the cell, and then it binds to the AR, forming this complex, in which it will translocate into the nucleus, and then it'll do two things, either two things, activate or repress target genes. It does this by various methods. You can see here it physically binds to the RNA, or it can bind to a transcription factor, some other type of complex. Either way, it regulates some kind of target gene control. So when we're looking at down-regulating AR, we have to use some kind of down-regulator, some kind of inhibitor. So I focus on traditional Chinese medicine and some compounds found in these remedies. Growing up, I was really exposed to my family's diet, and they heavily incorporate traditional Chinese medicine into their diet. And it uses a lot of rebel remedies to both treat and prevent diseases, cancer being one of them. So I saw this really huge gap in the cancer rates between China and US. And you can see here in this chart, China is over here. This uses ASRW, which is an age standardized rate. It basically takes into account an age gap so that you can easier compare these statistics. But you can see that in both prostate and breast cancer, US is way up here, all the way here, and then China is significantly lower. So there's this really huge gap, and it kind of adds to the idea that maybe natural products found in traditional Chinese medicine have this ability to prevent and treat cancer, in which downregulating AR in prostate and breast cancer can have a really vital role in treating and also a means of preventing cancer for the whole population. So I focus on apigenin. This is the compound I'm questioning. It is found in many different remedies, and these remedies have been shown to inhibit some kind of way for the AR function. And it was shown because it was efficient at delaying or prolonging the progression of prostate cancer. And so the goal is to validate this finding by experimenting on prostate cancer cells first, by treating them with apigenin, just to see if this is in fact the mechanism that it performs. And then if that is successful, because of the new novelty that AR is potentially connected to breast cancer, we will then expand with breast cancer cells later. So that leads to the hypothesis that apigenin will in fact bind to the receptor, such as how DHT, the natural ligand, does. And by doing so, it will in fact downregulate the expression of AR. I have here just a little bit of an experimental timeline. We began with researching all these natural products, a little bit about how traditional Chinese medicine works and the main compounds found, the active compounds found. And then we move on to computational analysis through this one software called MolSoft. Basically, I'm taking a look at the actual physical interaction and validating that there is some kind of interaction and some physical binding being involved. And then we're going to move on to prostate cells, where we take a look at treating these cells with apigenin. If that's successful, we will continue with the breast cancer cell lines. And then finally, we have results. So MolSoft. This is a computational software where it basically allowed me to have this chemical model of AR shown here. And then I'm using this to dock various compounds into the receptor. It takes into account a lot of things such as thermodynamics, chemical groups, steric hindrance to find a best fit area for this compound to bind. For here, we have DHT, which I said was the natural ligand, and it binds to this area, and it's called the ligand binding domain. So using this, we can measure the binding score of DHT, and I compared this binding score to all the different natural products that I analyzed to see which one is very similar to this natural ligand or potentially even better at binding. So I have here listed just all 10 compounds that I took a look. Again, they are found in many different traditional Chinese medicine remedies, such as herbal fats and everything. And we see here, apigenin had the most negative score. The more negative the score is, the stronger the physical binding interaction is. And you can see that it's actually a little bit more than DHT. So because of that, we can see that apigenin bound right here really nicely, very fittingly. And it also bound to the ligand binding domain. So the same domain as DHT. Furthermore, I took a look at the structural differences within DHT and apigenin. You can see that DHT is a steroid. Apigenin is not, but there are still some similarities. You see ring structures. Apigenin is a little bit more polar. But overall, you can see that there is some similarities, some differences. So that was really interesting to see. Moving forward, we're going to now take a look at the actual experiment with protein expression and if there is some downregulation of AR going on. So we begin with prostate cancer. And again, prostate cancer is really significant to the men because it is the 4th leading cause of cancer death in men. And because of this, AR is the main target for treatment. And we want to validate that apigenin, this natural product, can do something in terms of treating and preventing progression of cancer. So we test protein expression of specifically AR using these 3 compounds, DMSO. This is our vehicle solvent. Again, it's kind of the standard solvent to use because of stability. CA27, this is a synthetic analog to this one compound called curcumine. Basically, this compound has been found to, in fact, already down-regulate AR. So this is our positive control. And then we have our questioning compound, apigenin. And we use 50 micromolar concentration, so 0 and 50. And we use LinCap. This is our prostate cancer cell line that we're going to use for treatment. So after treating these cells with apigenin, I took a picture of the cells to take a look at cell morphology. And I'm not sure if you can see, but just as an overview, over here for DMSO, which is our negative control, it's quite spread out. Everything is laid flat, it's nicely confluent. And if you compare this with CA27, you can see in these regions, it's very clumped up. The cells are super stressed, it's balled up, and basically cell morphology did change. With apigenin, it's the same as DMSO where it's very nicely spread out, it's all flat, nothing happened. So we see that in CA27, something happened with cell morphology. And before, CA27 has been shown to be a little bit toxic to cells. And when using something as a treatment, you don't want it to be extremely toxic to the point where it can change cell morphology, because what if it impacts your healthy living cells? So we see here that apigenin didn't have any toxic effects to the cell morphology, so that was really good. So we continue with doing protein expression. I started with Bradford assay, which will just measure out complete protein concentration throughout the cells sample. And then we move on to SDS page that will separate out all proteins by size, and then transferring that onto a membrane, staining that membrane with Ponceau S. This will basically take a look at it validates our first step where we loaded these samples in. It takes a look at whether or not everything went well, where if everything was basically confluent and consistent. And then finally, Western blot, which will measure specific protein expression using specific antibodies against both AR and beta-actin. I have here beta-actin. This is what is called a housekeeping gene. This is a gene that is always active. It is constitutively active inside our body. So this basically is a positive control in analyzing our gel. It takes a look at, oh, something is happening in each well over here. So if you take a look at these results, we have AR on top. This is basically the size of AR on the gel. And you see with DMSO, we have A and trial B. There's a thick band at AR, meaning a lot of AR is going on. You see a lot of AR in this sample. With CA27, there's none. So there's no AR. It looks like there's almost complete downregulation of AR. Over here, apigenin, in both cases, it's right smack in the middle. So as I previously said, CA27 is the positive control. It is known to downregulate AR. But we have apigenin being quite close to the ability of CA27, where it did downregulate, just not as strongly as CA27, but it's still there. So we confirm that apigenin did successfully downregulate, at least upon treatment of these cells. So because we found that AR was downregulated by apigenin, we move on to breast cancer cells. We have a benign line first. It's called MCF10A. We have MCF7, which is a less aggressive breast cancer cell line. And then we have our triple negative breast cancer cell line. And again, we do the same experiment, but with triple negative breast cancer, it's novel because this is still upcoming research. Triple negative is the most aggressive breast cancer because it lacks all receptors. So it's hard for treatments to target something because there is no target. So if we find that there's this link to AR, that can potentially, increase treatment options. And we only treat these cell lines with apigenin, but at 0, 25, and 50 micromolar this time. I have another picture of these cells. Throughout these cell lines right here, here, and here, you see that the cells are basically the same before and after. At 0, it's the same. At 50, it's the same. So I didn't show 25 because it's just the middleman. But you can see that the cell morphology is the same, so it wasn't toxic. It didn't change anything. And then again, I continued with the same experiment where I took a look at the protein concentration, moved on with SDS page, stained the membrane, and then finally took a look at protein expression with Western blot. And on the right, I have the results. So first, I wanted to mention that AR was downregulated in 2 cell lines. We see it downregulated here and here. This is MCF7, and then this is MDA 453. So these are the cancer cell lines. So this is the non-aggressive one where at 0 apigenin, there's quite a thick band, and then it slowly progresses into a fainter band. So this is dose response. You can see that at 50, there's this huge band. At 25, it lowers. At 50, it's even more. The same thing happened with our triple negative breast cancer cell line, where at 0, there's this really thick band, and then it slowly progresses into a smaller band, showing that upon treatment of apigenin, there was downregulation. However, we see that it was not present, AR was not present in MCF10. This is our benign cell, benign breast epithelial cell line. You can see that there's just absolutely no band, but we know that something was going on because of our beta-actin. So this shows that MCF7 just doesn't express AR, and that kind of adds to the idea that potentially AR only plays a role in breast only when there is cancer occurrence. So that leads to the conclusion that because apigenin did successfully downregulate, as shown in the experiments, that it is a potential lead for drug development to downregulate AR for prostate and breast cancer cells, or breast cancer in general, for treatment and prevention. I have here just some references I used, and I wanted to do some acknowledgements. One for Dr. Makar Basafi for mentorship and just helping me throughout this whole project. Chapman University Schmid College for funds and support, and also CUE for the scholarly grant that actually allowed us to buy a new triple negative breast cancer cell line. Thank you very much. I'll now take. Yes.
How did you guys pick 50 micromolar for the concentration for exposure?
Yeah, so this is actually very high of a concentration. When doing something where you're wanting to confirm the mechanism of action or something, we want to see positive results. So we wanted to use a concentration high enough to initiate something so that we can visually see it and confirm. Because if we use something a little bit lower, it might not show up in our experiment. So we wanted to start off with a high concentration first to just test out if there is in fact this confirmed function going on.
I have a follow-up question.
Yeah.
So if you do that, would you be interested in doing future experiments where you do a more biologically relevant dose once you see if it actually works or not?
Yes, definitely. We wanted to expand onto that, but as you can see, we only got as far as 0, 25, and 50. Even at 25 micromolar, which is just 50% reduction, you saw some downregulation in the breast cancer cell lines. So if we kind of continue this, we could see some response at lower concentrations, but you would also take into effect that metabolism and everything going on physiologically inside your body, it might not correlate to these conditions. So there would be further research to be done. Yes.
So it sounds like apigenin might be a good target, as you concluded. So what kind of foods or where do you buy it, or is it something you can commercially buy?
It's not, because it's a natural product. So with using natural product, so first of all, to answer where apigenin is found, it's found in, like I said, a lot of herbal remedies, but I took a look and it's found in many regularly groceries. I think it's found in many vegetables, a lot of fruits like grapefruit. There's some teas that contain apigenin, and also it's a regular compound that is found in many plants. So there's that. And then it's not something that you can buy, because I'm not completely sure about this, but I believe that FDA doesn't approve for natural products as drugs because you can just find it in, like your everyday groceries. Usually they will approve of like synthetic drugs or something like that. So that's why epigenetic could be a lead compound for drug development. You can base off, from structure or just like the mechanism to target the cancer.
So where did you buy it to do your experiment?
We bought the chemical from ATCC. Yeah. Mhm. And then we just we mixed it with DMSO, our solvent vehicle.
Can you name one in the Chinese medicine that you were anything, the remedy that you use for the research?
It was a little bit broad. That was a little bit towards like the beginning of our research, but I think a lot of it was found in basically herbal teas. There was just so many that it was hard to choose, but I also personally researched it. It's found in chamomile. It's also found in this other one, but I'm not sure how to pronounce it in. I don't know how to say it in English, but I know it in
You can
Chinese.
speak in Chinese. You can
It's,
say this one in Chinese.
Dongguacha. Dongguacha.
Dongguacha.
Yeah.
Okay.
So I found that because my mom and I really love to discuss about this,
It's
but
winter melon tea.
yeah, it's found in winter melon. Yeah,
Winter melon tea.
it's found in like many different vegetables actually. So it's not just one. I believe grapes and celery has a lot of epigenin. But yeah, it's really interesting because I wanted to find what epigenin played a role as in these plants, like if it's like structural, what it plays. But it was kind of difficult to find what its role was. I'm just guessing it's a secondary metabolite, something that is just found within plants that maybe the function and like the role is just not known yet. Yeah. Right? Okay.
Let's take, as our next speaker is set up, I'll start introducing him. And so our next speaker is Cody Paresa. He is a biochemistry and molecular biology major and a health science minor. And his research advisor or mentor is Dr. Marco Bisaffi. And today he's going to talk about field cancerization of normal breast tissue through the use of exosomes of cancerous origin. Please welcome our speaker, Cody Paresa.
All right, hi everyone. I'm here to present to you on field cancerization in human breast tissue, and my mentor is Dr. Marco Bisaffi. So first of all, what is field cancerization? So the object behind field cancerization is that there's one of three conclusions or what it's thought to be. So first of all, you have phenotypically normal cells surrounding that eventually induce a tumor. That's the first thought. The second idea, which my project focuses on, is a tumor induces a field on the surrounding cells, making them act cancerous. And the third is a combination of the two. So as you can see, the shaded region represents the area of influence from the tumor or to the tumor. And this is going on at the molecular level, so you'll only see subtle changes in it, which is why I wanted to study this. So how do these fields act? So first of all, you can see that it'll be either homogeneous, so it'll have an even distribution on the surrounding cells, or the tumor can give rise and favor the formation in a single area over others. If you want to look at this image down here, this is a patient that was diagnosed with a lumpectomy. So they had a tumor in the breast. The tumor was removed, and then a pathologist would go in and check the margin of that tumor area. So they'll check 2 millimeters surrounding the area that was removed. And they would see, is there any cancerous cells in this 2 millimeter area? So once they were cleared from that, what ends up happening is 5 to 10 years down the line, about 30 to 45% of patients were having a reoccurrence of tumors, either in a primary tumor, so it'll occur in the same spot, which is in situ, or it'll occur in another spot, which is a secondary primary tumor, which is why I wanted to focus on this, because if the tumor was removed and they were cleared in the surrounding area, then how are they able to reform? So something is going on in those cells at the molecular level that we don't necessarily understand yet, which is why this is the focus of my project. So the problem here is that we don't really understand the environment surrounding cancer cells, which is why it's such a big issue. So we hypothesize that exosomes of cancerous origin are the major method of intracellular communication in the field of cancerization. So exosomes are seen to mediate cell proliferation and increase cell durability through the metabolic activity changes. So they were originally deemed to be junk vesicles. And why they were thought to be junk vesicles is because we didn't really understand what they did. So we're actually understanding that they have some form of intracellular communication, and they're actually upregulated in cancer cells. So as you can see here, you have your cancer cell. It's creating exosomes and communicating with a neighboring cell. Basically, that's the form of intracellular communication that we're trying to study. So how does this cancer cell impact the surrounding cells that are histologically normal? So our objective here is we want to provide evidence that our exosomes promote proliferation as well as cell viability. So how do we do this? First of all, we wanted to isolate our exosomes. So from our two cancer cell lines, we have our MCF7 cell line, which is representative of early stage cancer, and our MDA-MB453, which is representative of a metastatic cancer. And I'll get into those eventually. We wanted to determine the concentration of our exosomes through the use of a Bradford assay. We wanted to treat our healthy cell line, which is our MCF10A, with the cancerous exosomes to see the differences. We wanted to determine the viability of the cells using a trypan blue assay. And we also wanted to determine the metabolic activity of these cells using a WST assay. So first cell line is our MCF10A cell line. This was isolated from a 36-year-old patient. This is considered your literature normal cell line to use for breast cancer. The main thing here is I wanted you guys to focus on this because this is representative of the normal, histologically normal cell tissues for your patient. So, and then and also this is a naturally immortalized cell line, which means you can, for lab purposes, you can continuously grow and experiment on it. Our next cell line is our MCF7 cell line. It's our first cancerous cell line. It was isolated from a patient with an adenocarcinoma, which is a type of cancer. It's able to grow indefinitely because it is cancer. The patient was 69 years old. The main thing here is it's representative of early stage cancer, which is why we want to focus on it so much because so little is known about it. Our next cell line is the MDA-453 cancer cell line. It was isolated from a patient with a metastatic carcinoma. And again, it is able to grow indefinitely. But we just wanted to use this cell line, one, because we had it in the lab, and two, because it's representative of a metastatic cancer. So that's why we want to compare it to the early stage cancer and see, is there any difference between the two? So how do we isolate our exosomes? So first of all, you use a ultracentrifuge right here. And you start with a small amount of G's for a small amount of time. And then eventually you ramp up that. And eventually you're keeping the supername every time, but you first isolate your pellet, and your pellet is your cells. Then you'll isolate the pellet again, and you'll have dead cells, and then cell debris. And then eventually after that, you have a 2-hour process. Some people go the full 2 hours, some people go overnight. But if you think about it, you're leaving 100,000 G's overnight spinning, and just the amount of things that can go wrong is not good. So people typically stick to around 2 hours, but you can go less. But the longer it is, typically the more isolation we'll have. That's the main takeaway. So this is our Bradford assay. So we did this to determine the concentration of our protein. So you create this gradient right here. You take the average of the two and you create your standard curve. And then these are your samples that you want to test that are your isolated exosomes. And then you will plug in your absorbance value that you get for this into your Y and solve for your X. And then you will be able to determine your protein concentration of your exosomes. And this is done just to characterize it. Okay, we next did a trypan blue exclusion assay. This one was actually my favorite. So we use this to determine the viability of the cells. So first of all, what we did was we evenly plated 10,000 cells on a 24-well plate. And then we basically did a concentration gradient of a toxin introduced to it. Started with high concentration, eventually dropped that all the way down, just to kind of see how the concentration plays with the MCF10 cells, which is our normal cells. And then we also introduced a single concentration of exosomes to see no difference. The concentration in this case was 10 micromolars, but it depends on the amount that you're able to isolate from the previous process. So the main takeaway here I wanted you to see is this is kind of a simplified version of another experiment we did. This one was overnight. This one was 3 hours, but for some reason our control didn't work. There was too much cell death here. As you can see, the globs are stained cells. So how that works is we introduce a dye to it. And since the cells are dead, their membrane is permeable, so they are able to absorb the dye. And the healthy cells will be able to exclude that dye. So only the dead cells will have that dye in them. So we're able to basically remove the remaining dye, and then we have our cells right here plated, and then we can lyse the cells and measure at an absorbance of 590 nanometers. And then basically we're able to monitor the amount of cell death that we have. So it's a pretty cool concept. The main problem here with this result is that you do see our control had a lot of cell death, which kind of shows us that our toxin for some reason didn't work in this case, which is kind of weird. It was the opposite of what we expected. But you do see a proliferation with the addition of the exosomes, with both lines of exosomes, which is a good result. That's what we wanted to see. That was our hypothesis. We next did a WST assay. So this is based on the substrate to product conversion, and you're able to basically monitor the amount of product that is being made using a 96 well plate and a plate reader. And so for our WST assay, we measured at 440 nanometers, and we had 6 projects that we tried. So we did our control, our treated with our 2 exosomes, our toxin, which was sodium azide by itself, and with the addition of the MCF10A cells, and then our toxin with the addition of the exosomes. So in this experiment, you can see that there's no statistical significant difference between the addition of exosomes, which is not really what we wanted to see, but you can see that there is a difference with your control versus your addition of the toxin. So you see the toxin works in this case in regards to metabolic activity, but for some reason, our exosomes aren't working here, which is not really what we wanted. And then as you can see with the toxin and the exosomes, that there's not really a statistical significant difference here. So this is just a phenotypical approach I wanted you guys to see. So we have our control in the middle versus our one cancer versus the other cancer cell line. There's not really any visible phenotypical changes between the 3 images. Which is kind of expected because you're only expecting subtle differences with that. And it's all happening at the molecular level. So you would expect it a long ways down the line. You wouldn't expect it immediately. So this is an example of our control treated versus treated with the sodium azide. It's kind of hard to see on this picture, but you can see that the cell membranes are very permeable here. They're pretty weak. And you can also see gaps like such as this that don't exist in the control. So you can see that the sodium azide is again working at the phenotypical level and killing off the cells like we wanted. So we were able to isolate the exosomes and treat the MCF10A cells with the cancerous exosomes. We were able to use the Bradford assay for determining our concentration of the exosomes. We were able to use a trypan blue exclusion assay for showing the viability of the cells. The trypan blue showed that the toxin was ineffective, but it did show a positive effect from our exosomes, which is what we wanted. The WST for our metabolic assay showed that the exosomes had no metabolic effect, which is kind of weird again, but it did show a negative effect from the toxin, which is again what we wanted. Unfortunately, that means that we have inconclusive results at the moment, which leads us into our next steps. We want to improve our usage of the trypan blue assay, as well as the WST metabolic assay. We want to do dose and time dependent experiments with our exosomes. We want to try out different toxins instead of just using sodium azide. We want to use different cell models if possible. And also we want to do a more detailed characterization of our exosomes. Thank you. Any questions? Yes.
Why did you guys pick sodium azide to begin with?
So sodium azide is known to be super potent in the lab. It's super toxic, so we know that it would kill the cells. We didn't know what concentration to use because there's no literature values on it. That's why, as you can see earlier, here, we did a concentration gradient. So at the top, we actually had 10% and 5%, and it fully killed everything. Like there was nothing left, which is why we had to resort to the smaller concentrations.
Yeah, food for thought. Well, I use this in my lab and we use a really, really small amount of it and it does not kill the lab or did not kill the cells. So yeah, maybe it was just too concentrated and maybe that's why they were conflicting.
We did try other examples, like we tried formaldehyde, but it didn't really have an effect that we wanted to see. And that's kind of like when you're doing it, you're wasting like 2 weeks if it doesn't work. So it's like, like, do I want to work with this or not? But yeah. Yes.
The Bradford assay, right? It measures protein concentration.
Yes.
So what? Do you have any idea what proteins you're actually measuring in the exosomes?
So that's the thing with exosomes is they don't really know what it is with them yet, that because they still haven't been like fully characterized. So that's kind of what we were trying to see. We just know that they promote the proliferation of the cells. That's what we were trying to identify. And that's actually going to be one of the main focuses of my next semester because I am actually doing another semester here. So
I guess I have a follow-up question. So, only cancer cells produce exosomes or do?
No. Both do, but cancers are an exosome. Or sorry, they are an excess, produced in excess in cancer cell lines.
But other than that, are they similar? Is it known the exosomes that come out of healthy cells are the same as?
Yeah, so they're thought, again, to be junk, but they don't really know, and that's the thing. There's so little literature on it that's why I wanted to focus on it, because there's such little information out there. And if we were able to characterize that exosomes are the main thing that's causing this, that it'd be like groundbreaking idea. So that's why we wanted to focus on that. Any other questions?
Okay. Join me in thanking our speaker. Next speaker, as she's getting set up here, is my own student, Tiffany Lubrino. And I just want to say a few words. Also, I'm very happy that your parents made it. It's been a pleasure to have Tiffany in my lab. She's actually the first student who came and joined my group, and she just has an outstanding personality. She's very happy, very energetic, very enthusiastic. So I'm gonna miss that when she's gone. So please join me as she talks about one of the projects in my lab investigating the role of a protein called PGC1 alpha.
Thank you so much, Dr. Lopez. There you go.
Hey, everyone. Just for the sake of staying on time now, please welcome Tiffany Lubrino.
Good morning, everyone. I'm just gonna stand here since it's a different standing position. So hi, my name is Tiffany Lubrino. I'm a health science major, not Schmid, I'm Korean, but thank you again for Dr. Lopez for inviting me to the Schmid conference. And I will be presenting my research on investigating arginine methylation of, oh, okay, let's see. I think it's because I was too idle.
Got demethylated.
Luckily, it's still in the beginning. I will be my presentation on investigating arginine methylation of PGC1 alpha, a master metabolic regulator. So we'll first start with the table of contents. We'll have here from 1 to 6 from background, objectives, methods, results, as well as the results we did this semester in the spring, conclusions and future work, as well as acknowledgements. So we'll first start off with the background. Here in this figure right here shows the different family of PRMT family from PRMT1 through PRMT9. The main difference between each of these proteins are based on their particular protein structure, depending on where the methyl group deposits. So the one we'll be focusing on mainly for my presentation will be PRMT7, and its main one structure is MMA, versus the others that are ADMA versus SDMA. So what is PGC1 alpha? PGC1 alpha is known to be a peroxisome proliferator activated receptor gamma coactivator 1 alpha. It is known to be a transcriptional co-activator, and it's a very important role of mitochondrial biogenesis as well as energy levels. Here in this figure shows the full length 3D model of the PGC1 alpha. So how in PGC1 alpha is this helpful in terms of our body, and how does this affect our daily lifestyles? PGC1 alpha is implicated in the musculoskeletal system. Here is the lovely picture of a mitochondria. So we have a post-translational modification, such as you can do acetylation, methylation, phosphorylation, bind it onto the PGC1 alpha, and as a result will induce gene expression. And as a result, it would have specific varying mitochondrial levels. And how does this implicate in terms of the medical sense? So depending on whether PGC1 alpha is implicated in our bodies, it is a thermal regulator, as well as it will help in terms of diabetes and obesity regulation, and diabetes in terms for type 2. This protein is correlated with mitochondrial energy levels, as well as regulating the whole entire musculoskeletal system. PGC1 alpha is also heavily modified. Here in this figure shows the full length of PGC1 alpha. However, in this position, it shows a zoomed up portion of the C-terminus which is basically a small portion of what PGC1 alpha is. And then there are the specific residues that can be either phosphorylated, methylated, acetylated, ubiquitinated, etc. So what is our objectives? We have 3 main objectives. The first one, to determine the extent of arginine methylation of PGC1 alpha by our protein of interest, which is our PMT7. 2, mapping out the methylated arginine sites. And 3rd, determining whether methylation by PMT7 is temperature dependent or not. Here is our methods. But first, instead of using the full length of PGC1 alpha, we're only using a specific portion called that C-terminus, using it in 6 various temperatures, which I'll show later. This recombinant structure, like the piece of it that was given to us by Dr. Malk Stallcup from University of Southern California. The methylation using enzyme PRMT7 and our substrate PGC1 alpha as our 2 main primary focuses, as well as using that PGC1 alpha G1 protein. You mix it up with a little bit of the etomet, which is basically our methyl donor, incubate these reactions for 1 hour, and using that same reaction in 6 different temperatures of 37, 30, 21, 18, 16, and 4 degrees Celsius. We're going to go through a process of fluorography as well as mass spectrometry. 2 ways to detect them. First, through fluorography, like I mentioned before, you first detect the methylation proteins from that SES page by separating out the proteins based on molecular weight size. And once we find that specific portion is methylated, we cut out that specific portion and put it into what is known as mass spectrometry by identifying the methylated portions in which arginine residues are methylated. It goes through in-gel digestion, desalting using C18 stage tips, and process them in proteins. So here is our results. We go through a process, like I mentioned, fluorography, then Western blotting. So here is our results from fluorography. PGC1 alpha is methylated by PRMT7 at those 6 various temperatures. Here on this left side shows the fluorography as well as the Kamasi stain. Our no molecular ladder, we have G1 plus PRMT7, and then we have G1 alone as well as another lane with PRMT7 alone. Here on this right side figure shows the different combinations between PRMT7 and as well as our control, which is PRMT1, our substrate PGC1 alpha, and methylated donor etomet. We do a little combinations between each of the lanes, molecular ladder right here. But one thing to notice is the denoted methylated PGC1 alpha here, these asterisks, is around 38 to 39 kilodaltons. Here is a table for mass spectrometry results in these 6 different arginine temperatures. Here, for example, if you focus on 37 degrees as a little example, we have PRMT1 and PRMT7. Like I mentioned before, PRMT1 is our control. So let's say we focus this on arginine, the residue 548. We have different modifications, scores, confidence scores, sequence motifs. Here, PRMT1, the control, is also arginine methylated in both PRMT1 and PRMT7. However, if you compare it to 753, only PRMT1 is methylated at that specific residue, not PRMT7. In terms of the molecular modeling 3D model, here on this left side shows all the specific arginine methylated residues that were methylated at 548, 568, 570, and 753. Here on the right side shows the zoomed up portion where only 753 is heavily modified and methylated from PGC1 alpha by PRMT7. These figures were going through the process of 3D molecular modeling programs such as FIRE and PyMol. In terms of what we did so that was all in the past. How can we use all of this? Because in the past we did the full length. Let's do the same exact experiment, but this time, so sorry. In the past we did the one with just the C terminus. How can we do it in this semester by using the full length of PGC1 alpha? Since previous work was done only using the C terminus, will we get the similar results of full length PGC1 alpha at 2, instead of 6 specific temperatures, we only use 2. So the entire protein is inherently disordered where the arginine residues become inaccessible to PRMT7. Because using the full length PGC1 alpha, we decided to go through that process. We like to a little bit use an analogy of using like a string. A string is dynamic and changing, and it can move and dysmorphic in different shapes and lengths and whatever. And so how can we do that and explain it in terms of the full length protein instead of just using a part of the string? So this is the reason why we wanted to do the full length of it. And the protein model from this AlphaFold database used to reveal novel arginine residues. So same set of the experiment, just like before. This time we're just doing the full length. This time we used an anti-PGC1 alpha as well as anti-ADMA. We went through that same round and found out that the PRMT7 was mainly more methylated through anti-MMA. So we did that same experiment. Instead, for the primary antibodies, we use anti-ADMA. Here, right here in the left shows just a figure of how we created the sandwich, which is basically creating little fiber pads as well as the membrane and as well as the gel we use from SDS page. Put it in and then run it through, as well as a little figure. So here's our results showing here on anti-PGC1 alpha on the left and anti-MMA on the right side. We have the molecular weight as well as the PRMT plus PRMT7 plus G1, PRMT7 alone, and G1 alone. It's a little bit faint, but you can just see a little faint shown of the different bands that are between PRMT7 plus G1 and G1 alone. On the other side, for anti-MMA, you have the molecular weight ladder, and you do see 2 distinct bands for PRMT7 alone plus G1 and PRMT7 alone. So here is our conclusion. We figured out after completing all of these experiments, we found out that C terminus of PGC1 alpha were confirmed that this is a substrate for PRMT7, as well as optimized methylation, mainly at room temperatures from the range of 21 to 30 degrees Celsius. PRMT7 does methylate specific residue at 548 at all temperatures, and 548 and 568 are only methylated at 30 degrees Celsius. PGC1 alpha is also known to be temperature dependent, most likely allowing for greater functional control. So as we continue on, how does this role of arginine methylation of PGC1 alpha work in terms of the physiological setting? Like I mentioned before in earlier of our presentation, PGC1 alpha is known to be a thermal regulator and also used in cases such as type 2 diabetes and obesity. How can we implement this in terms of cell culture and using example of overregulation versus inhibiting ADOMET transferases and using this in terms of the cell culture state? So we have here in our future work for next semester, use the physiological role of arginine methylation, cell culture work, as well as HPLC work to detect arginine derivatives from cell lysates. So here on this right side, this is used from the HPLC. This is the this is here in the HPLC is what we use to compare cell lysates. So here is our amazing team. We have, as well as the amazing Dr. Lopez here in this room today. And then Paula, our research assistant, as well as the rest of our team, Sydney, Elizabeth, and Joshua. And then here's our acknowledgements. Thank you so much to the foremost members of the Zurita Lopez Lab at Cal State LA, the funding from the NIH, from the past, as well as the CUE grant. And thank you to my research mentor, Dr. Lopez, for all of our help. And these are the people who helped us in terms of HPLC training, cell culture training to Doris and Jill, as well as different Agilent technologies. And thank you to Schmid for this awesome conference. Well, concluding words, thanks to my family, Green mentors, Schmid mentors, pre-med mentors. Took this last week, but you're all the best. So thank you so much.
Questions for Tiffany?
Yes.
It's kind of impressive just how disordered PGC1 alpha is. Do you have any information on whether this post-translational modification changes the secondary structure, or if there are any other factors that influence it? For example, like when it binds to a target protein?
That's a good question. I can pull it up from here. So in this slide, here shows just only the primary structure for PGC1 alpha since it's the full length. We haven't gone into much literature detail in terms of how does the PTMs affect the secondary structure. But I think for only our literature review only focuses on the primary and full length. Yes.
Why did you guys originally select arginine as the target, basically, like as opposed to another amino acid?
Yeah, so a little background about this, the past work around here. So all of these results from here and these three, it was all from the work that Dr. Lopez's former students from Cal State LA chose in terms of arginine methylation. Also in Dr. Lopez's previous work, she did a lot of work in terms of arginine methylation here. And so we just tagged along and continued working from the previous institution to here at Chapman. Yes.
In its native state, is it a monomer?
So if you look for PGC1 alpha, I believe it's the, let's see. I actually do not know the answer to that question, but
Yes.
yeah, the answer is yes. Okay, yes, the answer is yes. That was a good question. I did not know the answer to. But yes, PGC1 alpha is a monomer in the native state. Yes.
I'm sorry if you mentioned this already, but what does methylation do for the protein? And also, like, do you plan on like expanding to know more about like what methylation does for the pathway or something?
Yeah, so we'll start here. PRMT7, the main one right here, because it's monomethylated. Right here in MMA, that's how we went through the Western blotting and the fluorography, find that specific methylation sites and figure out, okay, because in here, based on this specific structure of PRMT7, it is monomethylated. So if we went through the process of like arginine methylation, that's how we figured out, okay, this with methylation, it specifically targets helping thermoregulation and mitochondrial levels in terms of those two medical cases of diabetes and obesity. And that's how we found specific residues to find the spots of where arginine methylation is at that specific residue at that temperature.
Going and thinking, Tiffany.
So it's my great pleasure to introduce Tracie Okumura, who's been part of our group for about 2 years now. She's worked on several different projects relating to this enzyme here, fluorogenic acid esterase. We're going to talk about some of the most groundbreaking stuff that we results we got over the last 3 months or so. And fun fact about Tracie, that it's probably something everyone knows anyway. In addition to being a great scientist, as we all learned yesterday, she's an incredible second baseman.
Thank you, Dr. Owens. So like you said, my name is Tracie, and I will be talking to you all today about the structural and biochemical characterization of the chlorogenic acid esterase from the bacteria Lactobacillus helveticus. So let's take a few steps back and talk about what is chlorogenic acid, or CGA. So it's this beautiful molecule we have up here, and it's a component of plant and bacterial cell walls, which is why it's found in natural plant-based foods such as apples, carrots, coffee, and sunflower seeds. Chlorogenic acid, or CGA, has therapeutic effects. It acts as an anti-inflammatory and an anti-cancer agent. But in the same line, it also has undesired effects. So it gives coffee its really bitter flavor, and it also causes the greening of baked sunflower seed products. So how are we going to tackle these undesired effects? One possible solution is the enzyme chlorogenic acid esterases. These are industrially relevant enzymes produced by both bacteria and fungi, and they belong to the family alpha-beta hydrolases. So what they do basically is they add a water across this bond right here and cleave chlorogenic acid into caffeic acid and quinic acid. This allows it to eliminate those undesirable effects while maintaining those therapeutic effects. So the kinetics of that CGA hydrolysis reaction has been looked at in many different variations of this enzyme. Our lab studied Lactobacillus helveticus, which is in the second row, and its activity is so different from the other bacterial variant from Lactobacillus johnsonii. As you can see, its Vmax, or its reaction rate, is so much higher, and that's kind of our big question. We wonder why. Additionally, I wanted to add that compared to fungal esterases, Lactobacillus helveticus also has a much higher activity. Fungal esterases just have a really low activity in general. The question does remain, why are CGA's esterase activities so different? We wanted to look at the structure of these proteins to see if there are structural determinants of high enzyme activity. So on the left over here, I have the Lactobacillus johnsonii structure, which is, like I said, bacterial. And on the right here is the fungal variant from Aspergillus niger. They have fungal and bacterial enzymes have very different structures. So we kind of have a line of thought there. Maybe that's the reason for the difference in activity. But we want to focus mostly on the Lactobacillus CGA esterases. So we know Johnsonii's structure, but we don't know Helveticus' structure. So that's what we went with in our experimentation. So the first thing we had to do was clone Helveticus CGA esterase gene into E. coli, grow it, express it, and purify it. And right here is a gel where we have our purified protein at about 32 kilodaltons. And we use that purified protein in biophysical experiments to determine its properties in solution. And then we use crystallography techniques to solve its structure. So our first biophysical experiment was circular dichroism. So we basically use circular polarized light to generate the spectra in green of the folded protein, as well as the spectra in black of the heat denatured protein. The difference in the intensity between these two spectrums in terms of their molar ellipticity, which is basically how much the light is rotated, kind of told us that the protein is folded and that the folded spectra is what we expect to see from an alpha beta protein. So we additionally did a temperature ramp CD experiment where we increase the temperature, and as the temperature increases, the amount of unfolded protein also increases. And once we reach that halfway point where half of it is unfolded, that's known as our TM, or our melting point. And for Helveticus, that's 65 degrees Celsius, but denaturation does begin at 55 degrees. These properties are very similar to Lactobacillus johnsonii's properties. So structurally, they're both stable, and this is very similar. So we know that its stability isn't the reason for the difference in activity. So following those biophysical experiments, we set up crystal trays, we harvested the crystals, and then we collected several data sets to accumulate in this crystallography table right here. This data helps us evaluate if the crystal is good, the quality of the crystal, things like that. So I just wanted to point out a few key data points here. The first is the resolution, which was 2.2 angstroms. The second is the CC1/2 value, which is 0.979, the R-free value, which is 0.209, and lastly, the Ramachandran outliers made up 0.1% of the residues in our structure. Basically, this data is very good. This tells us that our structure is of very high quality. So this is said structure. What you see here is the functional unit of Lactobacillus helveticus in its homodimer form. So the homodimer form matches the other CGA esterases in terms of what's active. What you can see is this beta core surrounded by a bunch of alpha helices, and some of these alpha helices right over here, they make up the dimeric interaction that bring the two monomeric subunits together. There's a large surface of interaction in which there are salt bridges and hydrogen bonds that, like I said, bring the two monomers together. And here is said monomer. I isolated one of them and I colored it by its secondary structure. So this protein has 11 alpha helices and 12 beta strands. And of those beta strands, a majority of them make up this beta sheet that you see in the core. But here and there around the enzyme, you kind of see parallel and anti-parallel strand pairs. I do want to mention that in blue, I have highlighted the insertion domain, which is basically a structure that overhangs the active site of the enzyme, and it's unique to the bacterial versions of bacterial hydrolases, basically. So if you look at the fungal variants, they don't have this. Possibly that insertion domain makes the active site more specific to our substrate, but we have to do more experimentation to determine if that's true or not. So that brings me into kind of what structural determinants in Lactobacillus helveticus make it so active. So to kind of compare with what we know about Johnsonii, I did a structural alignment between the two, and it basically overlaid the structures in order to see the differences between them. So here we have Lactobacillus helveticus in pink and Johnsonii is in green. So you can kind of see that the structure is mostly conserved. So things are positioned the same, kind of in the same space, which is what we kind of expected to see because they're both bacterial, right? The main difference though is this loop that I've circled right here, which is in the Lactobacillus helveticus insertion domain, but it's not seen in Johnsonii's domain. This possibly can contribute to the difference in activity that we see, but we need to see how CGA fits into this active site in order to prove or disprove that hypothesis. So to do that, I use docking software, or we use docking software known as MoSoft to make these docking models that you see up here. So what MoSoft does is it takes CGA, it fits it into its active site, wiggles it around, and finds the most energetically favorable conformation. So again, helveticus is in pink and Johnsonii is in green. This is the surface view of each protein with CGA bound. The insertion domains that I mentioned that are unique to bacterial hydrolases are highlighted in blue, and the deep binding cleft of the active site are colored in yellow. There's kind of no obvious difference between the enzymes from this view, so I had to take a closer look at the active site. And so here is the helveticus CGA esterase active site. So I have CGA in green. This is our molecule, CGA. The protein is the pink, and I've just highlighted some of the important residues that I wanted you to look at here in blue. This is the same protein in both pictures. It's just kind of a different rotated view of it to see things a little bit differently, look at different residues. So the first thing that I wanted to point out is that there's a catalytic triad, which is found here, the aspartic acid 138, histidine 229, and serine 106. So basically there are acid-base interactions between these residues that allow them to perform a nucleophilic attack on CGA, which contributes to the hydrolysis or cleavage reaction. But the other thing that I wanted to point out about these pictures is that the majority of the intermolecular forces that we see are hydrogen bonds and salt bridges. I wanted to point out one in specific, which was with lysine 164 right here. This enzyme this residue is actually part of the loop that I mentioned that we see in helveticus, but not in Johnsonii. And the H bond that this residue forms is 2.2 angstroms long, which is actually a really strong H bond. So that's really exciting. So I'm going to show you a comparison in a moment, but that's something that I wanted you to know and remember. The third thing I wanted to talk about, was this tyrosine 173 residue, which is an aromatic amino acid, and it participates in pi stacking in a parallel conformation with CGA. So this is another way for CGA to, bind to the active site. And now we're going to take a look at Johnsonii. So some housekeeping first. This time, CGA is in pink, our protein is green, and the important residues are still blue. Again, both images show the same protein, just a little bit different. And I did want to note that I had rotated the view. So CGA binds these two enzymes in a very similar way. It's just shown like flipped in this picture. Okay, so again, this protein uses the catalytic triad in the same way. I won't point them out just because I showed you before. I did want to note that there is much more pi stacking in this active site, as you can see with tyrosine 169 and phenylalanine 160 in parallel conformation, and then phenylalanine 34 is in an orthogonal conformation with CGA. There are less H bonds and salt bridges than there were in Helveticus, but I did want to mention again the lysine 161 here. Of course, it's the sister molecule, corresponding to the other lysine I showed you, but this time it's 5 angstroms away from CGA. So that's a really big difference. So that might be something significant, and it may play a key role in the difference in activity. Lastly, I kind of wanted to talk about how Lactobacillus helveticus distorts CGA more than Johnsonii. So Lactobacillus helveticus is in blue and Johnsonii is in white. And this is basically just how CGA looks in the active site without the active site around it. The Johnsonii bond angle is 118 degrees, whereas the helveticus bond angle is 113 degrees. This bond is supposed to have a partial double bond character. So the closer we are to 120 degrees, the stronger and more stable that bond is. So since helveticus is further away, it means it has more single character and thus more flexibility. I also wanted to note that the dihedral angle on this side in Johnsonii is 20 degrees, whereas in helveticus, it's 28 degrees. A perfect double bond has a dihedral plane angle of 0. So the larger the angle, the more distortion we see, the more turning of the molecule. So basically, helveticus turns it more than Johnsonii does. This suggests that CGA is more contorted in helveticus, which is a classic case of ground state destabilization, which can lead to rate enhancement. However, these differences aren't really big or noticeable, so it's not 100% clear that this is the reason for the enhancement. And these are also from a docking model, which does give CGA more flexibility in kind of a very rudimentary way. And so in conclusion, some of the possible things that I mentioned are that, intermolecular forces that bind CGA to the active site might be responsible for the difference because they might distort CGA, or that lysine residue that I talked about may play a key role in the difference in activity since the bond lengths were so different. Ultimately, though, we are not 100% sure why helveticus is better from just the structure. It may actually be related to the dynamics of the protein, or basically how the protein moves as it does its work. And so for future directions, we want to crystallize helveticus CGA esterase while it's bound to CGA so that we don't have to use a docking model. There's no guessing involved, it's experimental values. And then we also wanted to do molecular dynamic simulations to, like I said, see how the protein moves. And then lastly, we can use this known structure to target residues in protein engineering and use site-direct immunogenesis to kind of knock out things in the active site and determine which residues are actually the key players. And that's all I have. Thank you so much. Yes.
A couple of questions. Wonderful talk. Thank you so much. That was intriguing. So you mentioned in situ mutagenesis for finding out which amino acids are doing what. But when you look at the sequences, the sequence differences between the proteins, could they be, outside of the active site, could they be the reason why one folds or changes CGA differently than the other?
Honestly, that's like another part of our project that we're definitely talking about. So we're actually looking at it to kind of improve activity. So we look at the B factor, which is the flexibility of each residue, and we're targeting ones that are the most flexible and thus have the best chance of taking in that mutation. And we are definitely not looking at the active site. We're looking everything away from the active site. So if we change one of those residues and the activity is enhanced, you'd be absolutely right. And a residue away from the active site would be part of the reason why the dynamics are so good. We haven't gotten too far in it yet, but it's exciting that's exactly where we're headed with our research too.
Can I have a second question?
Yeah, of course.
Thank you for answering that. You mentioned that you're expressing these proteins in E. coli. Is that a problem? As in, does E. coli do a correct job in expressing something that is originally not E. coli?
From what okay, so we initially started with 3 CGA esterases. Out of the 3, we stuck with Helveticus because it was giving us the best results, but one of them actually was mutated. Like, we expressed it and sent it back for sequencing and it was mutated. But what we've seen so far with Helveticus is that when it gets expressed, we send a little bit out of it for sequencing once in a while, and it comes back with the right sequence. So it seems like E. coli is doing a pretty good job of expressing the protein.
Excellent. Thank you so much.
Has anyone just mutated or got rid of the insertion domain, or has just studied the role of that domain?
I'm not too sure if it's been looked at too closely, but we do know that it's not in fungal esterases. So the overhang, this would be the insertion domain of the bacterial one. And I kind of tried to orient the fungal enzyme in the same way, and there's just nothing up there that would do the same job. And based on their activities, so it's kind of like, I would say almost like a natural mutation of not having the insertion domain. The fungal esterase activity is even lower than Johnsonii. It's incredibly low. So we're thinking, or I'm thinking at least, maybe the insertion domain does play a role in helping out the activity. So, but we haven't done any studies on eliminating it or seeing what it does specifically.
And presumably it would still be active without it.
Yes, because the fungal esterases don't have it and they still are active.
See none, let's thank the speaker one more time. So our second speaker of the session is Brendalyn Figueira. She has been part of the lab for about a year and a half, and like Tracie before her, she'll only be talking about really the most recent results, things she's been working on in the last few months. And I hope that maybe she'll be the first person in this, in this session to, instead of using a laser pointer, we'll be using one of those things to point.
You use those to dance.
And then maybe she'll be the first person on the session to use these to dance.
Well, I need someone to hold them, for I can dance in between them.
That floor yours.
Okay, so I will be talking about how we set out to characterize the interaction between CoW-N and MoVP, but with fluorescence. So a little background on nitrogenase. Nitrogenase serves as an essential role in nature that reduces atmospheric nitrogen into ammonia that is a usable product for plants. And this is in nitrogen fixation, which you can see in this equation right here. It's the one that does all of the work in order to get ammonia. And then the structure of nitrogenase is over here, and we can see that it's a 2-protein complex. So we have molybdenum iron protein right here that we call MoFeP, and then FeP is our iron protein that I'll call iron protein. And basically this enzyme does the reaction of reducing nitrogen gas into ammonia. And so what we know so far is that nitrogenase is inhibited by carbon monoxide. And we wanted to set out a bunch of different experiments to see how this protein, CoW-N, can protect this. So in this graph right here, we can see that we did, or Dr. Owens in our lab did many treatments of MoFeP and CoW-N alone and together, and in the presence of different concentrations of carbon monoxide. So you can see right here, the activity of the nitrogenase is very active with the presence of no carbon monoxide. And then when we add CoW-N into that, it's still able to do this. And once we add a higher amount of carbon monoxide at 0.1 ATM, the activity of nitrogenase completely diminishes. And then, but when we add CoW-N in it, there's a little activity change, and so it's somehow interacting with it. And so sort of like making it protected from the carbon monoxide. And then at an even lower concentration of carbon monoxide, we saw that CoN was actually almost able to fully recover the activity of nitrogenase. And so I wanted to understand the protein-protein interactions of this. And we have one piece of evidence that we can see that shows these interactions. And it's also with the specific activity that we used. So we can figure out with this graph that CoN actually is the protection is dose dependent. So as like on the x-axis is the amount of CoN we add, and with the amount of CoN increasing, the activity will increase as well. And this is fitting to a binding curve. So there's some sort of binding action that's happening. And we can see that it is dose dependent. And the KD, which is the apparent dissociation constant, it's really, really low, which is really good. And this is the apparent, what we think is the dissociation constant, but we haven't done an actual activity measuring the binding affinity. And so the research question that I wanted to go through was, well, first we know that CoN is able to project nitrogenase by lowering the carbon monoxide binding affinity. We know that they interact in some way, shape, or form, and the real KD between CoN and MoVP is unknown. So we wanted to measure it using a different way so that it's not measuring the activity of the enzyme, but rather the binding. And so I hypothesized that we can determine the binding affinity using fluorescence quenching. So one of the main things is I was like, how can I measure protein-protein interactions? And fluorescence was my answer. So these are the different ways that fluorescence can use, that can measure interactions. The first one being FRET, which is fluorescence resonance energy transfer. The next one is fluorescence quenching and fluorescence anisotropy and or polarization. And then I chose fluorescence quenching. So basically what quenching is the intermolecular deactivation that decreases the fluorescence intensity and the quantum yield. And as you can see in this diagram that I created, here, I'll move on. So basically there's going to be molecular contact between the fluorophore and the quencher. And this causes the fluorescence to completely decrease or like just lower a little bit. So we can see that quenching is proportional to binding. And when the light is shining on our protein that's just tagged with a fluorophore, it can fluoresce at a specific wavelength. And that is without this CoN and quencher present in the solution. But then once you add them together, when you shine light on it, the fluorophore actually transfers its energy to the quencher and the fluorescence is quenched. And you can see one of the things that I had to take account of is that the emission and absorption spectrum should overlap between the fluorophore and the quencher. So the quencher absorption is a little bit before, and it's this clear peak, and then the dye emission, which is the fluorophore, is a little bit shifted, but there is some spectral overlap. So these are the two proteins that I wanted to label, and we chose a thiol reactive fluorophore because MoVP actually has two cysteines on the surface of it, and with those cysteines, they have thiol reactive, or they have thiols. And we wanted a fluorophore that can bind onto these, so we chose a thiol reactive one. And then CoN, we wanted to label with the quencher, and it has a lysine residue on it, and with lysines, there's amines with it, so we chose an amine reactive quencher. And so with the labeling scheme, I basically decided for the fluorophore, we wanted to use rhodamine red C2 malonide, and with this, we predict it's going to attach onto that thiol group. And you can see in this this spectrum right here, it shows the wavelength at which the absorption is happening, and then the relative intensity of it. So this yellow peak right here would be the intensity of the fluorescence for this fluorophore. And then in this black dotted line, you can barely see it, but that is the spectrum overlap of the quencher. And the quencher that we used was QSY9 carboxylic acid succinyl ester. And this should react with the lysine. And this is showing our desalting column, which is basically, once we labeled it, we wanted to remove the excess label. So this bottom purple band is actually the bound label with the protein and the label bind it together. And then the top band is the unbound label. So the first thing that will elute into this little epitube and or it will elute and we'll have the protein. And then, so after labeling these proteins, we took the absorption of them, and we can see that with MoFeP labeled with V-fluorophore, there is a peak at the wavelength that we expected for this fluorophore. And then there's also a peak of the absorption of the quencher at the expected wavelength. So what we did was we did a fluorescence experiment using MoFeP, increasing the CalN, and then increasing CalN even more. And so we saw that quenching did happen. So this blue line right here shows the fluorescence peak of MoFeP on its own. It's fluorescing. This is what we wanted to see. And then, so this shows that there is, in this orange line, that's when we added CalN, but at like a very small concentration, only 0.26 micromolar. It is a 1-to-2 ratio of MoFeP and CalN. And there is some quenching that occurs. The fluorescence decreased, which is what we wanted to see. And, but we think that this is the full quenching ability because when we added an even more amount of CalN with the quencher, it was about the same fluorescence decrease. So there was no additional quenching, so there was perhaps too much added. But because there's quenching, there's some possible protein interaction, and that's what we wanted to see. And then, but unfortunately, we did our negative control of this, and basically we ran the same experiment, but instead of using the buffer that we used before, we used urea. And we wanted to use urea because we know that urea can unfold this protein. So with this, there shouldn't be any interactions and there shouldn't be quenching. And our negative control does show some quenching. So it's kind of giving us an inconclusive result, but we can see that quenching still does occur. And it actually, urea, the buffer that we used, increased the fluorescence. So that was interesting, but we saw quenching is occurring. And because the fluorescence is so much higher, it's in a dose-dependent way. So there's quenching still occurring, but urea did increase the signal. So we are doing some future directions with this. And one of the main things that I wanted to do was use a smaller concentration of CalN. And because we saw in that graph that the 1-to-2 in this yellow or in this orange is about the same as the 1-to-5. So we see that since they're a similar concentration, we wanted to have more of this dose-dependent thing that we've been seeing. So if we use smaller concentrations, we can see that more dose-dependent. And labeling different residues was also another option that we could use. And when we labeled different residues, we can find out like different fluorophore and quencher combinations to bind to the residues on the different proteins, and we can find better interactions or better like fluorescence signals. And then we also use a different negative control in the lab. We usually use oligomeric CalN, and this is a form of CalN that shouldn't be protecting MoFeP because it's unfolded. So it shouldn't even bind, and we shouldn't see any interactions. So thank you for listening, and I'd like to thank Dr. Owens and the rest of my lab. And here are my references.
How effective is the labeling?
So we were able to see that in these fluorescence, like this was right when we checked the absorptions after we labeled them. And basically when we wanted to see if it absorbed at a specific wavelength, and there is binding that we can see. And at this wavelength, it shows that there is an absorption at the fluorophore. So the fluorophore is bound to it in some way, shape, or form. And same with this, with CalN, it's at the peak is at the designated absorption spectrum, or the designated absorption like nanometer.
I guess I'll ask this in a different way. So like if you have 1 molar concentration of your protein, what % of that gets actually labeled with the fluorophore?
So when we were doing this experiment, we wanted to make sure that we had excess label. So we wanted to add a lot of label into it so that we can get as much binding as possible. And then after that, we were able to elute the different like unbound label. So we just tried to use an excess of label in order to properly bind, like allow it to bind. Like, because usually, like if I were to use like an equal amount, it would be 1/3 of it would bind. So we wanted to use the excess amount to see if more could bind. Okay. Any other? Yeah,
I have 7 questions.
7.
I have 2 questions. One is the fundamental one, and the other one is more technical. The technical I always ask, very nice data, by the way, very nicely presented. When you put a tag on a protein like you did, do you see any, do you expect any change to the, is there a chance that there was a change to the protein in terms of the structure?
That was one of the things that we were actually talking about in the lab. So when we were doing the labeling reaction of MoFeP specifically, because CalN is supposed to bind to it, there are those residues, similar residues in the active site. But we saw that like we didn't want it, give it enough time to weasel its way in. So we just decreased the amount of time that we had the reaction for in order to try and mitigate the binding that can happen. Because if it changed the active site, it wouldn't be able to bind. So since we did see some sort of interaction, we're seeing that there is some sort of binding that's happening or some sort of interaction.
I'm also asking because I saw that you used the 2 sticking out cysteines to bind something. I would assume that they are not engaged in any kind of disulfide bridge building. Otherwise, otherwise you would change the structure of that protein.
Yeah, and we chose those because those were on the surface and there wasn't anything near the active site that we know to, like, that we predict is what is being done to, in order to, that CalN is inhibiting because we don't, or not CalN, carbon monoxide is inhibiting. We want to make sure that it is not interacting. And we want to just make sure that carbon monoxide is not in there. So it does that answer your question?
Yes, it does. Yeah, it's fine.
Yeah.
The fundamental question I had, I'm not sure if this makes sense, but I'm not in this field. Carbon monoxide inhibits the enzyme.
Yes.
By binding to it.
Yes.
And then CalN somehow counteracts that.
Yes, because it should, what we presume is that it's binding in the active site of where carbon monoxide would be inhibiting.
So does it compete with carbon monoxide?
Yes.
Does carbon monoxide bind CalN?
No, we did not see that, but there is, where was it? So I don't remember where the slide is, but basically, could you repeat your question one more time?
Is carbon monoxide also binding CalN?
We,
in other words, does CalN act like a sponge for carbon monoxide so it doesn't inhibit the other enzyme?
So basically, we're thinking it's kind of like a lock and key mechanism. So it's like CalN should be going in to MoFeP in order to block the active site of carbon monoxide. And with carbon monoxide, like we can see at the high concentrations, we can see the high concentration of carbon monoxide, with CalN present, the activity is still able to happen. So because CalN is being added, then the more CalN that we add, the more protection that we can see, and it's still able to like do some sort of it's still able to do its process. The enzyme's still able to do its process when cowan is present and there is carbon monoxide.
Thank you.
So you mentioned labeling other amino acids. What other amino acids would you label?
So on this picture, so we know that there are two cysteines, but we wanted to, there's two cysteines on MoFeP and then lysine on the cow-N. And we know that there's another cysteine on cow-N's surface that we could possibly label. So that was another option. And we just wanted to see if there were different, I'm not sure what other amino acids that we could try, but we know that we can accurately label cysteine. That's why we wanted to choose it. We wanted to choose this one. But there are other residues that we can put a quencher on cow-N to see if it has any different interaction.
Is the criteria only that they have to be on the surface?
That's the ones that we've been focusing on because we don't want to go too deep in and change the function of the protein.
Right, you wouldn't want to label that right where how you assume cowan interacts with.
Yeah, so we have a presumed like active site and like what residues are in there and what residues would be interacting. So we want to use residues that wouldn't be interacting in the active site. But that's why, at least for the MoFeP, because there are some cysteines in that active site, we stopped the labeling reaction earlier so that it didn't have enough time to possibly label the active site. But any other questions?
Okay, I think we have to move on to the next speaker.
Yeah.
That's next speaker is Sophia Kelsey, who, despite what her badge says, is a biochemistry major. She's been part of our group for also about a year and a half. And we timed these presentations for our group here, because there's so many of us, we timed this order of presentations brilliantly, because Sophia is going to talk about how MoFeP and cowan interact and how that could be affecting cowan function, and answer all these questions that Dr. Rizzafi just had.
Yeah, see, I wasn't on that project.
The floor is yours.
Wrong.
Hi, thank you. My name is Sophia, and today I will be talking about characterizing the interactions between MoFeP and cowan using crystallography. So ammonia is an essential molecule used in biosynthesis, and however, it is very difficult to make ammonia since nitrogen in its gaseous state is very stable given its triple bond. And so currently industrial ammonia synthesis uses a Haber-Bosch process. However, this process uses very high pressure and also uses very high temperature. And this results in a lot of pollution and negative environmental impacts. Thus, it's really important for us to look for biological alternatives to reduce nitrogen gas into ammonia. So a biological alternative to the Haber-Bosch process is an enzyme called nitrogenase. Nitrogenase hydrolyzes ATP as a source of energy to make ammonia. And as seen in the equation below, it requires 16 ATP to break down dinitrogen into ammonia. So here's the structure of MoFeP, or of nitrogenase. Here at the top, you can see the iron protein, and at the bottom is the molybdenum iron protein, which I'll refer to as MoFeP. And there's also 3 metal clusters. So up here you have the 4 iron, 4 sulfur cluster. Here's the P cluster, and here's the FeMoco, which is the active site. At the top up here, the nucleotides is where ATP is hydrolyzed for the reaction to occur. So nitrogenase is inhibited by carbon monoxide. So on the right, you can see the FeMoco active site, as mentioned on the previous slide. And here you can see the purple is iron, the yellow is sulfur, the blue at the bottom is molybdenum. And then this red and gray complex is carbon monoxide. And here you can see that carbon monoxide enters into the active site here and displaces this yellow sulfur. And so that's how it is inhibiting nitrogenase activity. This exact mechanism is not 100% known. However, we do know that carbon monoxide comes into this active site to inhibit nitrogenase. So, however, even in the presence of carbon monoxide, nitrogenase is still active when another small protein called CoN is involved. As seen in the figure to the left here, under 1.1 ATM carbon monoxide, nitrogenase is fully inhibited. And under 0.001 ATM carbon monoxide, it's 50 to 60% inhibited. So you can see that here with this, there's no bar at all here, and here this is about 50 to 60% of its full activity over here. However, you can see when CoN is present in both of these situations, the activity can go back up. So here in high concentration of carbon monoxide, there's about a 9-fold change from this very, very, very low bar over here. And here the activity is almost fully brought back up to its activity in no carbon monoxide environment. So essentially CoN is a really important protein for the functioning of nitrogenase in the presence of carbon monoxide. So where does CoN bind? It is generally assumed that CoN provides a protection against carbon monoxide by interacting with MoFeP near the opening of the carbon monoxide access channel. So as you can see here, CoN can come in. So here's CoN, and it can come in through the access channel here to access the FeMoco and interact there to kind of block off that carbon monoxide, as a question Dr. Basafi was asking earlier. So that's kind of what is currently presumed happens. So my big question was, how does CoN interact with MoFeP to provide protection from carbon monoxide? So we wanted to get some real results and some real data so we could show that that is possibly what happened. So to do this, I used crystallography of the MoFeP and CoN complex to make a co-crystal structure. So here's the workflow of the process. We started out plating bacteria, growing it up, purifying the proteins, both CoN and MoFeP, and then the crystallography began. We set up the tray, monitored crystal growth, harvested crystals and froze them, performed X-ray diffraction, processed all of the data, and were able to solve crystal structures and analyze those structures. Today, I'll be mostly focusing on setting up the crystal tray and beyond, so the crystallography aspect of it once we have the proteins prepared. So some strategies we used to set up the crystal trays. The first strategy was basing the trays off of previously successful MoFeP crystals. So we wanted to try and use conditions we knew were successful in the past to see if we could co-crystallize CoN with MoFeP. The next strategy we tried to take was varying the CoN concentration and the CoN MoFeP ratio. And the last strategy was changing one component at a time. So along this crystal tray, we would change different concentrations of salt, a crowding agent, and pH. We tried to reduce salt concentrations because salt generally interacts with protein-protein interactions, kind of disrupts that. So we would want to reduce that to encourage CoN and MoFeP to come together in a complex. And with the crowding agent, that would be either MPD or PEG. And we would want to increase the concentration of the crowding agent to try and push those two proteins, CoN and MoFeP, together to try and force them to interact. And then lastly, we would change the pH buffer along either A through D or 1 through 6. So once we were able to grow crystals, our crystallography methods looked like this. So up here we have, this is a crystal in a loop. You can see like the brownish in the middle. That is actually a crystal in a really, really small loop. And from there, we were able to do X-ray diffraction. And this is one of the X-ray diffraction results we got. And from there, we used a variety of computer programs from IMOS-Film, Zaya2, Phaser, Phoenix, Qt, and Pymol. And we were able to get a final structure and search that for the presence of CoN. So this is an example of the crystallography data. On the left is a crystal table, and the statistics are based on the quality of the crystal. So basically, these statistics told us that we had solid crystals. So we looked at completeness being near 100%, and then the I over sigma value being 11.2 told us that there was a lot of signal and not a whole lot of noise. So these checked out, and we were able to say that we had good crystals. And on the right here, you can see an image of the MoFeP structure in Qt. So the yellow is the MoFeP structure, and then this blue grid-like map represents electron density. So what we did here is search around our big MoFeP structure to see if there was any excess electron density that was unexpected. So we searched around that structure, and here you can see, okay, this electron density is following the yellow structure very well. However, in the middle here, you can see there's kind of the spots of blue electron density that don't necessarily follow the structure. So that's kind of what we were looking for to see if that could be somewhere where Cowan was filling in. However, in this case of this image, this region is really small, so this is not necessarily somewhere where Cowan could be present, but that's what we were looking for when we were analyzing our structures. So the results from our search showed that there was no Cowan present. The crystallization of Cowan and MoFeP actually failed the first time. There were 3 solved structures that indicated that only MoFeP was crystallized. And on the right here, you can see one of the structures we solved, and that is only MoFeP. You can see the metal clusters in there as well. So a possible explanation for this is that Cowan, this small protein over here, could slide in between MoFeP. So on the right, this big colorful figure is the lattice of MoFeP when it lines up to make a crystal. So here you can see that there's these big gaps, like this one circled in red, and there's some here as well. And so Cowan, being relatively small in comparison to MoFeP, can slide in there and bind in more than one location and different orientations. So when that happens during X-ray diffraction, we're not able to get a diffraction pattern that shows Cowan in one spot over and over. So we're not able to see that clear crystal structure. And so that's one of the reasons that this may have failed. So to combat this, our next steps were to stabilize Cowan and MoFeP interaction using a diazurine crosslinker. So here we used a crosslinker to try and combine MoFeP and Cowan and kind of force them to interact so we could get the co-crystal structure. So at the top here, you can see this is the diazurine crosslinker, and at the bottom here is the final structure of MoFeP, crosslinker, and then Cowan. So this is the mechanism of how this works. The crosslinker binds to a lysine on MoFeP and then forms a diradical which interacts with Cowan. So once we had this final structure, the goal was to crystallize this complex. So far, there are no results with the diazurine crosslinker. And in the future, we want to continue moving forward with this crosslinking. So trying various conditions. For example, we want to try and crosslink first and then concentrate the protein down for our experiments so we can see, did this cross-linking actually work before we put it in the crystal drop? Or we could try and cross-link within the crystal drop. However, we're not entirely sure how cross-linking works at such high concentrations. So that might be something to trial and error with. And then lastly, does unreacted Cowan or cross-linker need to be removed? We don't know if these molecules being left in the crystal drop would interact with the formation of a solid lattice. So they may need to be removed, which would prevent us from doing it inside the drop as well. And then lastly, in the future, we're going to start experiments using N-truncated Cowan. And so N-truncated Cowan is Cowan as seen here on the right with this N-terminus region cut off. So the N-terminus region is quite disordered and sticks out from that more main 4 helical bundle that you can see. And so if we are able to cut that off and repeat some of the crystallography experiments and try and get Cowan and MoFeP together, we could see if that N-terminus region did in fact impact the formation of a clear crystal lattice. So that is where we are headed in the future, and some of those experiments are underway. So here's my citations, and thank you so much to Dr. Owens and my whole lab team for making this possible, and also just the constant support through the past years. Yeah, thank you. Any questions? Yes, it is.
So how do you know that the cross-linker is going to bind to the same way that it would biologically?
That's actually a question that I have thought about throughout this. And so we don't know that it would work in the exact same way, but it's kind of a foot in the door in like cross-linking and trying to make sure we can get a crystal structure. So solving this crystal structure and seeing where those interact would kind of be our first steps. And from there saying, okay, is this somewhere that's like biologically relevant? Would this make sense? And then moving forward with that and trying to continue to co-crystallize them without the crosslinker. Yeah.
Sorry, I missed it. Maybe I missed it, but what's the original evidence that Cowan and that protein MoFeP interact? What paper or?
So right now we have this. This is one of the main pieces of evidence we have that Cowan provides protection. So we can see just based off of when Cowan is present, like the activity can go back up. So we know that there's like some sort of interaction against the carbon monoxide to protect it. Yes.
Yeah, I had an additional question. Thank you, Davis, for asking that question. That was one of mine, important one. Fundamentally, can you crystallize cow N by itself? And then you have the crystal structures for those two individual, and then use some sort of, I don't know, MolSoft or something to see how they interact? Would that be a valid approach?
That's a good question. I'm not entirely sure what the state of crystallizing cow-N is. I hopped on this project when we began the co-crystallizing process, and I know that the structure of cow-N has kind of been a little bit of back and forth kind of on what that final structure looks like. Here's a better one. You can see the agreed upon structure of cow-N, but I'm not sure we have tried to crystallize just cow-N in the lab. But that is definitely something to think about in the future, and if we could do that. I think over the summer, Dustin possibly worked on some sort of experiment where they were docking.
Yeah, I mean, fundamentally, it's not the same as we all understand as biochemists, that when two things interact, there is structural change.
Mm-hmm.
But since it seems to be a challenge to co-crystallize them together.
Yeah.
Thank you.
See, yeah, we're a little bit early, so we have time for more questions. Sophia?
I'll ask one more question. Is it a 1-to-1 ratio? So it's 1 cow-N or 1?
So that's what that's kind of what we currently assume with it coming in and blocking that access channel here. However, we did kind of play around with different ratios, not knowing exactly if it is that 1-to-1 ratio, and having more cow-N either to encourage more binding or just to see if somehow there are two involved. So we have played around with the concentration kind of without results so far.
Is carbon monoxide something that is found at these concentrations in nature? You know?
I do not know necessarily what concentration carbon monoxide is found in nature.
But obviously it's there.
Yeah, it's present in some way, just in the air. Where is that's true. Here you can see carbon monoxide does inhibit quite a bit, either at the low concentration or the high concentration. So kind of whatever concentration it is at, it has some sort of effect. So that's kind of where we went with this.
That's a huge difference there. That's 100.
It is a pretty big difference between none at all and 50 to 60%.
I'm talking about the concentrations of.
Oh yeah, the concentration difference pretty big.
It's 100 times.
So anywhere between there, we would assume is would be somewhere in this range, which would still be a lot of inhibition.
With that, let's thank the speaker. It's my pleasure to introduce Caroline Monahan. She's been part of my group for about, was it a bit over a year?
Just a year.
A year or so. And she's been very busy developing a fluorescence assay that allows us to characterize esterase mutants. So if you remember, Tracie talked about this protein a little while ago. This is basically the follow-up to that work.
Yeah, thanks for that introduction. Yes, I'm Caroline. If you don't know me, I'm part of Owen's esterase team. I thought what better way to present my work than to look like my work. That's why I'm wearing the sweater today. But I'm going to be, like Owen said, talking about my developing my fluorescence assay for the rapid screening of esterase mutants. So some context, which I feel like is always beneficial, is I'm going to introduce you to chlorogenic acid, or CGA. So CGA can be found in sunflower meal, which is a byproduct of sunflower oil production. And in alkaline conditions, when CGA interacts with amino acids via its quinone component, it causes this greening effect, which makes it just turns it green. Doesn't really change like the taste. Like if we turned the sunflower meal to sunflower flour, it'd be fine, but people don't want to eat green cookies, I guess. So it's currently just discarded. And we would like to prevent that because food waste is bad and it can be used for a gluten-free alternative to flour, which would be a good benefit to society. So the Asterase team has worked long and hard and has been successful at degreening this, or like stopping this greening effect via the hydrolysis of CGA through esterase enzymes, but it's not perfect yet. So going into esterases, we're working with bacterial CGA esterases, and like I said, it works quite good. We're pretty successful in the degreening process, but it's not as efficient as it could be, and we could just do like a little, like we could just fine tune this a little bit better to achieve complete degreening. And so if you look at this graph, which is a Michaelis Menten graph, you can see that there's not a huge difference between the Vmax at low and high temperatures. The Vmax is just, it shows like the rate of reaction basically, or the like it represents the maximum rate of reaction. So if we can increase this, then we can increase the efficiency and effectiveness of the esterase enzyme. So we are going to engineer CGA esterases in order to increase its utility for food science applications. So how are we going to do this? Our long-term plans are, step one is to crystallize to visualize esterase, such as my associate Tracie was talking about earlier. After we got our 3D model, we then use B factor analysis to determine where to mutate, which my associate Kelly has been working on. We found 10 mutations. And then from there, we're going to be generating these mutations via PCR. We're going to screen the mutants by a rapid assay, what I'm working on. And then we're going to characterize our best mutants and then rinse and repeat. Going into a little bit about B factor analysis. So B factors are values that indicate disorder in a protein. A high B factor indicates that there's disorder in the protein structure, so it's more flexible. And high B factors are like I said, are found in flexible regions. And it's been found that like mutating these regions, these B factor residues, like it will improve the enzyme activity. So we determined 10 target residues for mutations. And each of the colors on this CG esterase structure represent like a different area, a different like residue. So how are we going to make these mutations? We're using site-directed mutagenesis, or SDM. Basically, this method uses specific primers to target a single point mutation. And then we're going to be using PCR to create copies of these mutants. We'll treat them with KLD and transform them into competent cells and then grow the colonies. It's important to note that we don't know what the specific mutation is when we're using these mutants. We'll only know what the mutation is once we send the mutant off to sequencing. So that's like, so basically we're going to be, it's kind of like a blind test, and then we're going to see if it works well. If it works well, like if we find this one that, oh my God, this one works really great, we're going to send that one off to sequencing and figure out what mutation was. So that's leading us into our problem. So our current method for screening enzyme activity is pretty time consuming. Our method is HPLC, which the analysis of the samples can only be done one at a time, and it also requires an additional step to clean up the samples. Like I said, we're doing 10 mutations. So if we did like 10 mutations and made 10 mutants from each of those mutations, then we'll have 100 mutants. And then let's say we'll do like quadruplets, that's 400 samples that we need to like analyze. And doing that one at a time would just take years. I gotta graduate. So our solution is to develop a fluorescence assay to rapidly screen all of these mutants. So in case you don't know what fluorescence is, I'm going to go through a quick 101 on fluorescence. Basically, fluorescence occurs when a fluorophore absorbs light, gets excited, enters the excited state, and then when it drops from the first excited state back down to the ground state, it emits a light, and fluorescence measures that emission of light. And then what's a fluorophore? So not all molecules will fluoresce. Only molecules containing aromatic groups or conjugated systems are going to be capable of fluorescing. So in this case, CGA, and then its hydrolysis product, or like it's when it's broken down, caffeic acid, is also a fluorophore. So let's go into some assay development. Woohoo! So first, in order to develop the assay, we needed to pick an excitation wavelength. In picking this wavelength, we wanted to make sure that there was going to be a differentiation between CGA and CA. And then we also wanted to make sure that we weren't going to be fluorescing any DNA or proteins, because under 300 nanometers, those molecules will fluoresce. And if you want to make sure that we're just tracking CGA and CA, it's good to not have a bunch of other messy fluorescent signals getting in the way. So we landed on 392 nanometers as our excitation wavelength. And then we determined the optimal buffer to use. So we found that by increasing the viscosity of the buffer, we increased our fluorescent signal. So we're adding 10% glycerol to our heaps buffer, and we're just going to be using that for the remainder of our experiments. And then we finally got a tracked hydrolysis to identify trends using just the standard spec. So going into that, using the standard spec, we found that there was a blue shift in the spectra after hydrolysis. So from there, we realized that CGA and CA are fluorescing at different wavelengths. CGA fluoresces at 490 nanometers, while caffeic acid fluoresces at 470 nanometers. And this is just important because this means that we can we can track the reaction. If we see that blue shift in the spectrum, then we can assume that the hydrolysis was successful and the reaction went to completion. Now going into another trend that we saw. So when we were doing hydrolysis, we noticed that there was a decrease in fluorescent signal after hydrolysis had occurred. And we didn't know why this was happening, because if we put CA and CGA alone, not using our enzyme, like as you can see, CA, which is the blue, which is like the blue line, has a much higher signal than CGA. But after hydrolysis, we were finding that there was a much lower signal, and we didn't know why that was. And we found that QA, the second product of hydrolysis of CGA, was acting as a quencher molecule. So as you can see, with the addition of QA, the blue peak here, which is CA, goes down to the gray line here, which is a huge difference in fluorescent signal. And then we can see the same for CGA when we added quinic acid to it. And I just want to note that we also mixed like CGA and CA together to make sure that CGA wasn't a possible quencher, and it wasn't. So we know that effectively that QA is. And this is doesn't really matter, but it's really important that we can explain this when tracking reaction. And yeah. So going into our proof of principle test, we wanted to make sure that our assay was feasible. So we performed a proof of principle test with a wild type. Oh, I also meant to note, going back for our assay, this is all done with the standard spec. And then when we started doing a rapid using it for mutants, we started using a 96-well plate. So we were growing our expression cultures on our 96-well plate. We're using the plate reader to do our fluorescence assay. And we made some slight adjustments in order to use a 96-well plate. So everything else is done in a 96-well plate. So for our proof of principle test, like I was saying, in the graph I showed you, we were expecting a decrease in fluorescent signal. So in this graph here, we were expecting to see the same trend with our wild type, and we did. We can see that after CJ hydrolysis, there was a shift and a decrease in fluorescent signal. So cool, that's awesome. And then looking at our SDS page gel, we were expecting to induce expression. So this shows that the induced cells have CG esterase, which means we induce expression. And then the uninduced cells, we do not see this line. So we can know that the expression was not induced. So now I got to do the exciting thing of doing this whole process from start to finish by making my own mutant. So I picked H245 mutant to test. It is our only mutant that has, it is the only core residue with a high B factor. And it is this yellow region right here. So I used our PCR primers to make our mutant and produce copies. I did our KLD treatment. I grew the colonies and our 96-well plate, and we induced expression. And our next steps means I get to start using my assay to check the activity as well as perform our Bradford assay. And then I can analyze all of the results, and we can see if this mutant is possibly something that we can work with further in the future. So just going over the whole workflow one more time, we're going to be making our random point mutation from the B factor, from the residues that we determined from our B factor analysis. Produce copies via PCR, treat with KLD, grow our colonies. We're gonna grow the cells overnight and express, and then screen for activity using the fluorescence assay, and then perform a Bradford assay. And just acknowledgements. A huge thank you to Dr. Owens and Christine for being some great mentors, and the rest of the S-Rays team that has made this possible. And here are my some sources. And thanks. I open the floor to any questions.
Yeah, I have a couple of questions. That was very interesting. Thank you for that. Caffeic acid, is that caffeine?
No.
It's not. Is that not the same? So you're not making coffee?
I wish I was making coffee.
It's always exciting to be involved in developing an acid.
Really makes you feel like a mad scientist.
Right.
Like, what am I gonna try today?
Make sure that you're gonna be on the patent. It's my recommendation. So that was interesting. I didn't really get the information well about the IDP. Actually, the B factor. The B factor signals or represents. Is that the same as intrinsically disordered protein sequences? What is the B factor and why is it connected to the mutants?
Well, B factors are just like, let me go back.
All of these.
So,
They were good.
There we are. So they're just like B factors are just like a value that will indicate like order or disorder. So if you like look at this specific value and if you look at this region and it has a really high B factor, then we know that there's like high disorder in the protein. So perhaps I don't know for certain, but perhaps that could be like an area that is like what you just mentioned. Sorry, I'm blanking on the name. But it's just a value that represents areas that are like more flexible or have more disorder. So we just know that we could probably like improve that area via.
So, and those are the residues that you're targeting by introducing mutations, right?
Yeah.
And you're looking for an enzyme that works better.
Yeah, we want to like mutate it so it will be like much better because in other like I said, like our enzyme our enzyme is pretty great, but like more related related esterases that like other researchers have been using, like they have they're just like working much faster and better than ours, and so like we just want to we want to have the best esterase on the market. So
thank you for that. Yeah, makes sense.
Any other questions?
Can I see the paper airplane transition?
Yeah.
Yes. That was good.
That's fun on PowerPoint.
Can I just ask a follow-up questions about it's a very potent assay, introducing one mutation at a time. Is that what you're doing? One mutation at a time?
You were targeting a point, like 1 point, but like when we do the PCR, it makes like all the stuff at once, like all the different mutations.
But is it always only one amino acid that is changed, or can you introduce two changes at the same time?
Oh, I'm not completely sure. I know that we use specific primers, so I'm assuming that the primers are just specific to like a one site mutation and not like a multiple site. But Dr. Owens might know.
Because I keep thinking maybe a combination of mutant amino acids might do the trick.
Oh, well, that's like the rinse and repeat is kind of like once we see like, oh, this mutation did well, like we might be able to like now like sequence that, take that one and be like, okay, where else can we mutate on this? And maybe they'll be like make it better.
It's like a synergistic situation. Very interesting.
A lot of fine tuning.
Hear none, let's thank Caroline. So with that, we are at our final speaker of today's session. The speakers Emily Wong. Emily Wong has been part of our group for about 20 years maybe. I mean, it feels like that. No, she's been part of our group for 3 years. She's really done a lot of work, a lot of excellent work. She's already on a publication, and most likely will be on a second one based on this current work that she's going to present. Again, like all the speakers today, the nitrogenous speakers especially, this is sort of the more recent stuff. The stuff she did over the last year or so, right?
Roughly.
Roughly a year. So with that, Emily, the floor is yours.
Thank you for the introduction, Dr. Owens. So, hello, my name is Emily Wong. I'm a part of Dr. Owens' structural biology lab, and today I'm going to be talking about how does CalN bind to nitrogenase. So before I dive into our experiments, I'm going to take a little bit of step back and give you an introduction. So nitrogen itself is a very useful compound. In its most common form, N2 or dinitrogen, it's very stable. So in order for it to be used by plants, it needs to be converted into ammonia. Now on an industrial scale, this process is done via the Haber-Bosch process, which requires a lot of resources such as high heat, high pressure, and fossil fuels, none of which are actually good for the environment. So there is an alternative to this process, and this is via the protein nitrogenase, which is expressed in the bacteria Glucosidobacter diazotrophicus, or GD. So nitrogenase is able to undergo this nitrogen fixation process by taking dinitrogen plus energy in the form of ATP and then converting it into ammonia as seen in this equation below. So nitrogenase is made up of two subunits, an iron protein subunit and a molybdenum iron protein subunit. And its active site is made up of the P cluster and the FUMOCO clusters, both of which are, well, metal clusters contained within nitrogenase. So the thing about nitrogenase is that it is inhibited by carbon monoxide. So under conditions containing carbon monoxide, nitrogenase will be unable to reduce substrates. So we characterize this protein called CalN, which seems to protect nitrogenase in some way. So to test this, we initially ran an activity assay using acetylene gas, and this measures enzyme activity based on the amount of acetylene that is able to be reduced into a substrate, in this case, ethylene. So we start with our control of nitrogenase in no carbon monoxide, and we see relatively high amounts. We see pretty much high amounts of activity. When we put it at 0.1 atmospheres of carbon monoxide, we see this activity be completely reduced. This means that nitrogenase is inhibited. So then when we put it at 1,000th of an atmosphere of carbon monoxide, which is what we would consider what you would find in the general atmosphere, we see nitrogenase activity be reduced to roughly 50%. So what happens when we add CalN into this mix? At the higher concentration of carbon monoxide, the nitrogenase activity is brought to about 20%. But at the 1,000th of an atmosphere, or the lower concentration, nitrogenase activity is completely restored. So we conclude that CalN does have a protective role in and is able to keep nitrogenase active even when carbon monoxide is present. Now, not shown, we've also ran some other experiments that show that nitrogenase and CalN actually do interact with each other. So this leads us to our question. So as of right now, CalN is predicted to be a 4 helical bundle, as seen in this figure over here. Now, I say predicted because as of right now, we don't really have much experimental data, and crystallography for CalN is currently unsuccessful so far. So therefore, we don't know how CalN works. So our research question is to determine which part of CalN is important for its function and therefore for nitrogenase binding. And we hypothesized that by changing a critical portion of CalN, the protective abilities of CalN will thereby be affected. So how do we go about determining what are these critical portions? We first started off with a sequence alignment, and this is where we take multiple CalN species and just line up all the sequences and compare them next to each other. Over here, I have 4 different CalN species, with RGD being highlighted in the star. Now, note, I'm only showing 4. The original alignment was actually done with many more CalN sequences. So what we concluded out of this is that the C-terminus of CalN contains various conserved sequences across several diazotrof species. The 2 that I want to point out is a glutamic acid seen in the 87 position, or denoted by E, and a cysteine in the 90 position, denoted by a C. And we hypothesized that these conserved sequences may play a major role in CalN function. So a series of mutants were then created to determine how these mutations would affect the structure of CalN, as well as its ability to protect nitrogenase from carbon monoxide. And by mutating these residues within the C-terminus, we can hypothesize if there are any specific residues that may play a significant role in proper binding and or function. Now, to my left, I have another figure of CalN that just shows where we plan on mutating the amino acids, visualized over here. And to the left of that is a table showing just our various mutants. So these mutants we're changing its physical properties, such as our glutamic acid changing to an alanine. Some of these are a little bit more subtle, such as our cysteine to a serine. And we also wanted to make a mutant as radical as possible. In our case, truncating our C-terminus, which is just removing the last 2 residues of CalN. So to determine how these mutants would affect the activity of nitrogenase, we ran an activity assay that is very similar to the one initially ran to determine how CalN would affect nitrogenase. Like that first one, this is an ethylene turnover assay, which will measure acetylene reduction to ethylene gas via gas chromatography, and the amount of ethylene that's produced. The amount of ethylene produced is what's going to determine how much enzyme activity there is. So we start off with our control of no carbon monoxide and no CalN, and we see activity levels at roughly around 800. So then when we introduce our wild type CalN and carbon monoxide, we see activity levels hovering closer to 300. And we also have a negative control, which is our oligomeric CalN. Oligomeric being this is going to be our misfolded CalN, and we know this is not going to protect nitrogenase. And this is true as in we see activity levels at around less than 100. So then as we work our way through our mutants, we start with E87A. So E87A has low activity, very similar to that of the oligomeric, which we can conclude that E87A is unable to protect nitrogenase. Meanwhile, E87Q actually has activity levels that's higher than that of the wild type, which we can conclude out of this that this is able to protect nitrogenase. As we go with our C90S and C90A mutants, both of these have activity levels that is very similar to that of wild type CalN, meaning that both of these mutants are able to protect nitrogenase. M91A and C truncated even, both have activity levels that are very low, just like the oligomeric, meaning both of these mutants are unable to protect nitrogenase. So what we conclude out of this activity assay is that the C-terminus is important for CalN structure and function. Although based on the activity assay alone, we don't know if this is a structural thing or if this is due to amino acid characteristics. So in order to further investigate why these mutants may be inactive, we ran a CD spectroscopy. So CD spectroscopy, or circular dichroism, utilizes circularized polarized light to measure the secondary structures of polar molecules, or in our case, proteins. So the signal shape and intensity will determine how much folding there is. The stronger the signals, the more folding there are in the proteins. Likewise, the weaker the signals, the less folding there is. So I actually split these mutants up into 2 categories. The first one I'm going to show you is our folded CalN. As when we're looking at the CD spec compared to our wild type, all of our folded CalNs is going to be E87A, C90A, and C90S. Now, all of these actually had very strong signals, meaning that they were able to maintain their secondary structure, and they were able to maintain the characteristics of their alpha helical folds, which this makes sense when for C90A and C90S, this makes sense when we're comparing it to the activity assay I showed you before, as since these two mutants were folded, they were able to protect nitrogenase. But what is interesting is that E87A is actually folded, and E87A is one of the mutants that is folded, but on the activity assay, it was inactive. So this just goes to show that just because one of the CalN is folded, just because CalN is inactive, that doesn't necessarily mean that is unfolded. Now, the reason I say this is when we look at the next two mutants, are M91A and C truncated. Both of these have weaker signals, and therefore means that they lost their secondary structures and they're unfolded and also unstable. Now, the M91A and C truncated were both mutants that were inactive on the activity assay, which means they're unable to protect nitrogenase. Based on the CD spectra, this makes sense because if they're unfolded, they can't protect nitrogenase from carbon monoxide. So what can we conclude out of this? So structural implications, the C-terminus plays a significant role in proper folding and function of CalN, and M91 is important for folding. Now, both of these were shown based on the CD spec from before, since these two mutants were the ones that were unfolded. On the mechanistic side, C90 is not critical for CalN's protective function. Now, the reason for this is unknown, as in the original sequence alignment, C90 was one of the most conserved residues across all the CalN species. So in the future, we plan on investigating on why this is the case. What we did find is that E87 is important for function. And we propose that the reason for this is that E87 is able to form a salt bridge with nitrogenase. So glutamic acid is inherently polar and will more likely form ionic bonds. Some of these bonds being salt bridges. So we'll further test this if E87 is able to interact with nitrogenase using future binding experiments such as cross-linking, which in short is adding a functional group on the protein and forcing an interaction between them. These are just some of my references that I use. And special thanks to Dr. Owens, my mentor and advisor throughout the entire experience, Christine Laverde and Dustin Willard for providing me advice during this process, all of my lab mates, and for all of the viewers today for attending and viewing our results. Thank you, and do you have any questions?
Floor is open for questions.
I have some
Yes.
questions. Very, very nicely presented.
Thank you.
If you can go back to that list, to that one slide where you show the mutations that you, yes. So I was wondering, is there any rationale in choosing the mutations? Like, for example, why, maybe you said that and I missed it. Why change a 90 into an alanine and not, I don't know, a glutamic acid? I don't know whether that makes sense, but is there a rationale for the choice of the mutated amino acid?
There is. So I can start off with you explaining the reason behind cysteine. So cysteine, it will form like disulfide bonds. So the reason why we want to mutate it into a serine is it's good for the cysteine to serine mutants. That's going to maintain the same size. The molecule is going to stay the same size, but you're going to lose the disulfide bond. When we change for alanine, that's actually going to that's going to not only change the size, but that's also going to remove all the properties. That's also a similar case for the glutamic acid to glutamine and the glutamic acid to the alanine. Similar reasoning to the glutamine. For glutamic acid to glutamine, that's maintaining similar properties, but it's a little bit more subtle compared to the glutamic acid to our alanine. So it's pretty much we're just trying to changing it, but also in some ways we're keeping it some similarities.
So this is the opposite of being a random choice.
Yes.
Rationale.
Yes.
Okay. Can I also ask, this little protein seems to be a challenge for crystallization.
It is, yes.
And I always thought that the smaller the protein, the easier it is to crystallize something. I'm not a crystallographer, so I don't know whether that's true or not. The CD measurements. Can you go there?
Would you like the folded or the unfolded?
Doesn't matter.
Okay.
You said that this shows the level of folding of a particular protein.
Correct, yes.
Okay, is this specific for any kind of folding, or is it just folding?
I believe this is just like for folding, and we're primarily looking at the secondary structures. So it's just
And is it specific for specific secondary structures?
based on the CD spectra alone, or
Yes. Like when you look at this data, can you say this is beta sheets, this is alpha helices, this is, I don't know, beta triplets?
yes, you can actually tell from based on this whether this is beta sheets or alpha helices. In our case, we are looking specifically more towards this region, which will tell us the alpha helices, since Cowan is predicted to be a 4 alpha helical bundle.
That's a prediction.
Yes. And
It's not a proven.
currently not proven, but there's been enough. This prediction was based off of like multiple Cowan species, so we can pretty much say that Cowan are 4 alpha helices.
Thank you.
Any more questions?
Can I ask one more
Yeah.
fundamental question?
Sure.
Plenty of time.
Cow N.
Keep asking.
What induces cow N?
Ooh, now that I'm not too sure about.
Could it be carbon monoxide itself?
I mean, it's a possible theory. As of right now, all we know about Cowan is that it's most likely a lock. As mentioned earlier, it's a lock and key with nitrogenase. Although, again, since Cowan there's still a lot to be studied, that is a possibility.
And you mentioned that there are many different sequences, right?
Yes.
You only showed 4 or 5. Or
Yes, there's a lot.
So each organism has its own cow N variant.
Yes.
It's interesting. Thank you.
Okay, if there are no more questions, let's thank Emily one more time and let's thank